Good afternoon, everyone. I just want to make sure that I'm audible.
We can hear you.
Yes, thank you.
Thank you. So we are Group 7 from SAPRA. My name is Esmeralda Cordier, and our group will be discussing the key differences between the regulatory assessment of biosimilars and that of a novel biological medicine. Next slide. So the various elements in the presentation are outlined in this slide. We will not be reading through these questions as it's incorporated in the slides. Sorry. Okay, so a biosimilar is a biological medicine that's highly similar to another already approved biological medicine, the reference medicine. Biosimilars are not considered generic versions of biological medicines, as the molecular microheterogeneity cannot be precisely replicated due to the natural variability and more complex manufacturing processes of biological medicines. So for the purpose of this presentation, we will be incorporating examples based on a monoclonal antibody trastuzumab, of which the reference medicine is Herceptin and the biosimilar Ontruzant, and we'll be using the term product and medicine interchangeably in the presentation. So concerning the regulatory assessment of a biosimilar versus a novel biological medicine, for the latter, full clinical, preclinical, and quality studies are needed to demonstrate safety, efficacy, and quality. But for biosimilars, comparative quality, non-clinical and clinical studies, which includes mainly PK/PD studies, and confirmatory studies are required to demonstrate that the biosimilar has similar safety, efficacy, and immunogenicity to that of the reference product. So the importance is to show no clinical meaningful differences from the reference medicine. Okay, so the next two slides illustrates the data requirements of a biosimilar and a reference medicine, as well as the comparative studies mentioned in the previous slide. So next slide, please. And next one. So the following key elements can be considered in terms of the product profile: product name and classification, of which some information was provided earlier. And then in terms of the active substance, the Ontruzant EPAR indicated that laboratory studies revealed high similarity to Herceptin in terms of structure, purity, and biological activity. And then the mechanism of action is to bind to the HER2 receptor on cancer cells and blocking its activity, breast and gastric cancers. Then approved therapeutic indications. So for both products, it's indicated for EBC, MBC, and metastatic gastric cancer. Next slide. The recommended dose and route of administration, it's the same for both. And then in terms of the manufacturing process, the cell lines or organisms utilized, the description of the production procedures, and a control of critical quality attributes that relate to the manufacturing processes can be looked at. And the stability and storage. So for both products, the shelf life of unopened vials, it's 4 years, and it should be stored between 2 to 8 degrees Celsius. Next slide. Pharmacodynamics and pharmacokinetics.
Claudia, can I please ask everyone to mute their mics? We're getting some background noise. Thank you. Go ahead.
Thank you. So for pharmacodynamics and pharmacokinetics, it includes absorption, distribution, metabolism, and excretion, onset and duration of action, and biomarkers to monitor effectiveness. In terms of clinical studies, so 10 main clinical studies were conducted for Herceptin across the 3 indications, and 1 study combining EBC and MBC to determine the effectiveness of Ontruzant versus Herceptin. It was concluded that Ontruzant would behave the same as Herceptin. And then comparability, it's applicable to biosimilars. It includes a comparison with the reference medicine as mentioned previously. And lastly, regulatory information refers to the approval status in different jurisdictions and the risk classification and legal category. Next slide. In terms of CMC activities, it ensures that medicines are consistently manufactured and meet quality standards. It applies throughout the product development lifecycle and help ensure that quality standards for purity, strength, identity, and stability are met. The criticality of different elements in a biosimilar CMC file is assessed based on their impact on the quality, safety, and efficacy of the product. It's important to understand a product because appropriate control relies on an overall strategy guided by product understanding. The quality attributes should be identified and ranked as key characteristics of the molecule, known as critical quality attributes, by their risk of impacting product safety and efficacy. In concerning manufacturing, critical process parameters should be identified, adequately monitored and controlled, and it's just the steps in the manufacturing process that can influence the critical quality attributes. Analytical methods to assess product quality should be able to detect variations between the biosimilar and the reference medicine, as well as to assess stability studies. And in terms of safety and immunogenicity, the impact of product and process-related impurities on a patient's safety should be assessed, as well as the likelihood of immune responses due to structural differences or impurities. So the CMC activities can aid improving biosimilarity between the biosimilar and the reference medicine. Thank you.
My name
Good day,
is
everyone.
Dominique Tusaho. Good afternoon, everyone. So I'll be, okay.
Good day, everyone. My name is Duduzile. I will be presenting the role of critical quality attributes in novel biological medicines and biosimilar medicines. So with regards to novel biological medicines, it's to identify and define critical quality attributes to develop novel biological medicines. Also to optimize manufacturing, quality, and clinical outcomes of novel biological medicines, to establish and ensure safety and efficacy, to establish critical process parameters to control manufacturing process and maintain batch-to-batch consistency, also to a detailed characterization of critical quality attributes to approve novel biological medicines. With regards to biosimilar medicines, it's to demonstrate comparability to the reference medicine, which is the biosimilarity. The manufacturing process is reverse engineered and optimized. Also to monitor and minimize variability between biosimilar and the reference medicine. Critical quality attributes analyzed to determine clinically significant of observed differences between the reference medicine and biosimilar. It's critical to evaluate the reference medicine in depth in order to establish or categorize the quality target product profile. Also to allow for extrapolation of clinical indication of the reference medicine to the biosimilar. Also comprehensive clinical quality attributes data within a specified range required to ensure biosimilars meet stringent quality standards to obtain biosimilarity approval. Thank you.
So my name is Dominique Tusaho. I'm continuing with where my colleague has left off. I will be discussing the relevance of clinical testing in biosimilar development. My focus will be to distinct between healthy volunteers and patients. So in a healthy volunteer, the focus is on pharmacokinetics, maximum concentration, and pharmacodynamic to demonstrate biosimilarity. It's normally conducted on a smaller sample size during initial testing phase. Generally, no treatment response are measured, and it's highly sensitive, so there is no comorbidities or concomitant therapy there. And it determines the ethical consent for biological medicine with significant side effect. So in patients, once the PK and PD is established, the focus is mainly on clinical efficacy and safety. It's used to confirm indication and perform immunogenicity testing. So normally this one, when it's conducted in patients, it's conducted on a target population with a larger sample sizes. And it's used to assess symptoms improvement or disease control, or it monitors adverse events and immunogenicity. This one is more expensive to conduct in patients and is also time consuming. It produces variable results due to the inclusion of comorbidities and concomitant therapies that are included. It provides evidence or justification for extrapolation of indication. So the position of the Medicine and Health Product Regulatory Agency of the United Kingdom on clinical testing for biosimilars. So biosimilar development and comparability. So these biosimilars should be should closely resemble the reference medicine in physicochemical property, biological activity, and clinical profiles. So the difference in impurity profile and experience must be justified for safety and efficacy. The amino acid sequence of the active substance should remain the same except for post-translational modification. The posology and administration route of a biosimilar must be the same as those of the reference medical product, but deviations from the reference medical product are possible, such as strength, formulation, excipients, or presentation. So this will require justification and may need additional data. So here we look at the confirmatory pharmacokinetic trials that are conducted. So when these trials, the PK trials, when they are conducted, they should be able to demonstrate equivalence to the reference medicine, preferably in healthy volunteers, as discussed before. If they fail the PK study or they fail, the root cause analysis should be provided and conclusion reflected in a subsequent study. So those results should not just be discarded off. So the PD pharmacodynamic parameters can be measured during the trial, and descriptive results should be presented. Safety and immunogenicity data should be collected and presented during the trial. So when confirmatory efficacy trials are conducted, so this confirmatory trial, normally they are developed and should be confirmatory efficacy trial. So when biosimilar development is done, it should be evaluated on case-to-case basis. So it's not done on all the studies. And a comparative efficacy trial may not be necessary if the scientific rationale supports it. A well-argued justification for the absence of an efficacy trial should be added to the clinical trial declarations. So there are principles for justifications. So those principles include the following: justification for comparable efficacy, for comparable safety and immunogenicity, and the need for efficacy or safety trial. So if the indication needs to be claimed for the biosimilar candidate, the following should be considered. The biosimilar candidate can claim the reference medical product indication if it is highly similar to the reference medicine. Indication can be claimed without further justification unless it is protected by market exclusivity or patent. So another topic that we were assigned to do was to discuss our position as Group 7 with regards to the use of reliance to UK approved biosimilars as compared to those of SAPRA, since our group, all of us were employees of SAPRA. So here SAPRA has decided to harmonize certain SAPRA medicine policies and procedures with those of EMA, and it has adopted a reliance review approach as a pathway for registration of medicine. So this approach allows the authority to leverage evaluation effort done by a recognized regulatory authority named as RRAs that it aligns itself with. This makes the evaluation process more efficient and enhance market access, since this is one of the policies of SAPRA. So for the PI and the pill content, SAPRA uses a reliance whenever it's possible, as stipulated in their SAPRA reliance guideline. And then the MHRA UK is one of the RRAs that SAPRA aligns itself with. So the principle that SAPRA use in terms of a reliance-based evaluation one of those principles is that they are using average review. When this one is conducted, is primary based on a primary, is primary based on reviews of preclinical and clinical data in the CTD. And then all the chemical entities, the biological application and generic application with clinical data that have prior approval from an RRA will be considered for an average review. So this is the statement on the SAPRA policy. So biosimilar medicine will be considered for average review, which is indicated specifically for the following types of applications. So these are the conditions. Number one, the biologic medicine must be registered by a recognized regulatory agency that SAPRA aligns itself with. Secondly, the biosimilar medicine where the reference product medicine has already been registered with SAPRA.
Good afternoon. My name is Corlia Kruger, and I will be presenting this slide that answers the question, in which cases do you think a patient study is essential, and when do you think a patient trial could be waived? So patient studies are essential in biosimilar development when challenges are experienced in obtaining unredacted assessment reports from the reference regulatory agencies, when public assessment reports lack the necessary decision-making skills, when a case requires a comparative efficacy or safety trial due to lack of understanding of biological functions or inadequate characterization of critical quality attributes that were covered in the previous slides. And also when safety uncertainties cannot be resolved without patient exposure pre-licensing, which is the approval or registration of the product. Patient trials can be waived in resource-limited settings when resource-saving benefits of using reliance-based review practices would be highly beneficial. Also in cases where the biosimilar pathology and administration route matches the reference medicine product. Exceptions or deviations may be required for strength, pharmaceutical formulation, excipients, or presentation. Ideally, the same reference product should be used throughout the comparability program. A different reference product may be used in clinical trials when these are sourced from a country that utilizes similar scientific and regulatory standards. So the handling of different indications for the biosimilar product in relation to the indications of the reference product. Regulatory agencies are mainly responsible for deciding whether to allow the applicability of data for different indications. There is a need to set up appropriate regulatory oversight for the licensing and post-market surveillance of biosimilars for all approved indications. Rigorous guidelines are required to mitigate the risks associated with the extrapolation of indications for biosimilars. This includes a comprehensive stepwise evaluation of the biosimilar's similarity to the reference product across multiple domains, including quality, safety, and efficacy. This systematic evaluation ensures that the extrapolation of indications is scientifically justified and supported by substantial data, demonstrating that the biosimilar will perform similarly to the reference product in all approved indications. In addition to pre-approval studies, continuous real-world evidence collection plays a critical role in supporting the safe and effective use of biosimilars across different indications. Post-marketing surveillance and pharmacovigilance activities are essential to monitor the long-term safety and efficacy of biosimilars as they are used in diverse patient populations. Real-world evidence generated from these activities provides ongoing insights into the performance of biosimilars in clinical practice, allowing for the detection of any issues that were not apparent in pre-approval trials. The SAPRA guideline for biosimilars state that there must be evidence for efficacy and safety for each indication. Then this slide just provides the criteria to be used for indication extrapolation. Extrapolation of indications for biosimilars. Extrapolation is a concept that permits the approval of a biosimilar for use in some or all therapeutic indications of the reference medicine not directly evaluated in clinical studies of the biosimilar. Extrapolation is based on robust scientific data generated in comparability studies and the totality of evidence. The concept of extrapolation is based on all available data and information in the biosimilar application, previous findings of safety and efficacy for other approved indications of the reference product, and knowledge and consideration of various scientific factors for each indication. Indications for biosimilars that demonstrate biosimilarity to the reference medicine in terms of quality, safety, and efficacy are generally approved. Regulators may approve all indications by extrapolation based on the data submitted for only one indication, provided that scientific justification is provided.
Okay, so this is what we've learned as a group, as Group 7. We've learned that bridged review pathways to evaluate biosimilar medicines is valuable for regulatory authorities with limited resources. Biosimilars are highly similar to the reference medicine and must show no clinically meaningful differences in safety, efficacy, and immunogenicity from the reference medicine. Biosimilars are approved through analytical studies, clinical comparisons, and PK testing rather than full clinical trials like the reference medicine. Extrapolation, authorizing biosimilars for therapeutic indications that have not been studied directly but supported by the same mechanism of action. Interchangeability, biosimilars are more affordable than the reference medicine and can improve patient access. The same reference medicine should be used throughout the comparability program. However, with the aim of facilitating the global development of biosimilars and to avoid unnecessary repetition of clinical trials, it may be possible to use a different reference medicine in the clinical trial or trials. This will need to be authorized in and sourced from a country with a similar scientific and regulatory standards approved by an RRA. Thank you. And these are the sources that we've used to compile our presentation. Thank you.
Thank you very much for that presentation, which brings now all together all the ideas that have been discussed before. Lise will have to leave very soon, but maybe she's still around to assist with one or two questions. And then I would like also to hand it over again to Arnold for further discussion.
Thank you very much, Bernd. And thank you to the group for the interesting presentation. I very much enjoyed it. Also your conclusions and reflections in the end. So thank you for that. I indeed have one question for you, which is perhaps a bit of a different question than I asked to the other groups. As regulators, you have an important role in evaluating medicines for their safety and efficacy with regards to biosimilar evaluation to evaluate that a biosimilar has high comparability against the reference product. But besides this, as a regulator or regulatory authority, you have also a role in informing patients, healthcare professionals about use of these type of medicines. And what we observed in Europe was that many patients and healthcare professionals had questions and doubts about the safety of biosimilars. And they had some questions with regards to the concept of what now a biosimilarity demonstration is. And I was wondering in case a patient or a healthcare professional would approach you about biosimilars and they would express concern, what would be for you some key messages or key elements that you would like to share with them to reassure them about the scientific rigor that underpins biosimilar development and evaluation? Is there anyone of the group who would like to give it a start?
Yes, I would like to give it a start. As a pharmacist, I would advise the doctor or the patient that the quality was established and it was vigorously analyzed. And if it is a biosimilar, then I will refer to the safety profile of the reference product, indicating that it has been evaluated for, if it has been patented for 10 years, there will be 10 years worth of safety or pharmacovigilance data available. So I would use that approach, but my colleagues can add on to if they want to.
So in addition, we can also assure them that the difference between the reference product and the biosimilar is in their molecular structure, and then that it is sometimes it's impossible to replicate some of this biologic complex molecule, but replication is not possible, but similarities has been demonstrated between the two products. So hence, when the reference okay, so when the biosimilars when we approve the biosimilar, it takes the indications for the biologic product. Thank you.
Lisa, I know you have to go, so you are excused. I'd like to follow up a bit because
Yes,
Dominica.
Just to say goodbye to everyone. Thank you for the presentations, and I leave you in the safe hands of Arnold. Thank you very much, everyone. Bye-bye.
Hey, have a good day, Lisa.
Really. Bye-bye.
I'd like to follow up, Dominica, a little bit on what you were saying, because you are using words that are not helping doctors and patients. That's the problem. And you talk about similarity and other things. So we have learned now to simplify the message to doctors and patients by saying the biosimilar product is structurally like the molecule in all essential parts where the messages are the same. And the resulting clinical profile is not different from the originator. So talking about variations, variability, and all the other things, it's just confusing. We see that in the US where they muddled up the whole thing when in the substitution discussion. So you have to simplify the message very clear. The molecule in essential parts is the same and it's doing the same. And that's the science base of drug action. Are there other?
Can I just make one additional point, and that was a message to healthcare professionals. And of course, then the message to patients would need to be even more simplified,
Yes.
which sounds easy, but that the devil sits in the details. So that can become very difficult.
Okay.
And it's a science in itself.
What we have learned in addition to that, Bernd, is that the whole chain has to talk to patient with the same language. So the prescriber, the pharmacist, the nurse, they all should express trust in the biosimilar. If one of these people in the chain say, well, yeah, actually, I don't know because they were telling that they are similar, but I don't know how similar, etc., that is raising suspicion among patients. First of all, I would like to make some final comments. I don't have any questions because I think this was an excellent presentation and I'm very happy with it. My compliments for the whole team, and especially because you showed that you all worked on it and you presented each your own part. So thank you very much for that. In the meantime, I had a little chat with Professor Samsalek, and I would like to stress, and that is for all groups that are working on biosimilars. Drug assessment should be science-based. That's the whole process. And we know that countries have guidelines, but some of these guidelines, they are old. SAFRA has recently changed the guidelines. So the last presentation showed how SAFRA has updated the guidelines and now are critically looking at the necessity of clinical trials. They allow reliance and etc. And I hope that the representatives and the students from other assessment agencies understand that essentially that the biosimilar should be the same as a reference product. So you should ask the same questions. So it doesn't make sense to ask additional questions that are not science-based on a biosimilar that you did not ask at a reference product. So one of the people was saying, well, we have to look at the dossier of the reference product, what is there, because we don't have to ask that again for the biosimilar. We look at similarity, we don't look at additional data. So it is about the evidence for similarity. And I just would like to make a plea. Don't ask for these additional things like stability data or ethnic difference, etc., if there is no science basis, because the result will be you won't have biosimilars. Your market in most countries is too small and they do not warrant the expenses and the efforts for these additional studies, and just companies will back out. I hope you don't mind that I made this comment because I think it's very important that we are aware of our responsibilities, and otherwise the patients in the end will suffer. I hope you don't mind that I made this comment, Bernd, and that you agree, and then I will give the floor back to Professor Rosenkrantz.
Yeah, thank you very much. I think that is a extremely very valuable point, and that is also part of the messages, I think, that we need to take into account. And talking about science, science is evolving. So it's just up to the state of the art at the moment that the regulatory submission is being reviewed, and that there may be some guidelines are actually outdated. That's actually the reason that the FDA guidelines, they all have the title draft guidelines to make it clear that these are not final documents that have to be adhered to like the law, but that they are matter of discussion and debate, even at the point in time of the discussion of the regulatory submissions. That's why we need well-trained regulators. We are so happy to have so many and young, enthusiastic regulators now around the team here, and just carry the can and take that message forward.
Final question, Sam. Have I addressed your point now, Sam Salek? Have I addressed your point adequately in this way?
Absolutely. Thank you so much, Arnold. That was amazing. And, you know, we really need the help of the assessors to take the biosimilars to a different level, because we are not there with the biosimilar where the promises that was made, 15, 20 years ago. So some of the comments that you made, absolutely on the spot. Thank you so much.
Thank you, Sam. Well,
thank you. And it concludes the day for today. And we would like to wish all of you a successful week again. For the groups that are going to present again next week, have a look again at your presentations that you plan. And maybe also, because these will be biosimilars again, specific examples, if there's anything that you learned now during the day today, these 2 hours, that may change your presentation, feel free to do that. And then we are all looking forward to the interesting discussions again next week on Thursday. Bye-bye and all best wishes.
Thank you so much, Bert.
thank you very much, and compliments for all the
Thank
students.
you.
Thank you. Thank you.